So without further ado, I will let Ed show us the path.
Great. Thank you, Colbert. Thanks. As Colbert so graciously introduced me, I've actually been at Charles River for 26 years. I am a local person. I grew up in the West Island and actually got a job very nearby out in Centerville, not knowing that it would kind of open a gateway to eventual global reach in the countries, 7 countries around the world and 20 plus sites, getting to know a lot of people in a lot of different cultures, all focused on innovative research, helping drugs get to patients faster in a safe way and making an impact on human lives. So when I first started, we were about 600 people at the Centerville location. We didn't talk a lot about what we did because some of the work that we do involves animal research, and there are certain parts of society that have strong feelings about some of that work in different ways. But we've gotten much better as we've advanced and become more of a partner in drug development, at telling our story, at talking about how we impact patient lives, animal lives, and really have an important touch to society. And from a great research institution like we're here at today, I think has a good appreciation of the work that goes into making that kind of impact. So I thought I'd tell a little bit of our story and some of the innovative things that we've been working on as part of our strategy within the safety assessment business of Charles River. So feel free to ask any questions. I know there's a Q&A session at the end. I like to keep things pretty informal and pretty light. So please do bring anything forward that you've got. So a little bit of our global reach for the safety assessment business, and maybe I'll start there. Charles River, most of you in research may know us as a vendor of mice and rats used in research. And actually, as we were doing a bit of prep for this session, as I started to talk about some of the things that we do a little bit more scientifically in our safety assessment business, even those in the field think of us as mice and rats and a vendor. And in fact, a couple of years ago, I went to McGill and was doing a job fair and was with their research faculty. And someone came up and said, I need to ask you about my order of mice. And it's actually not a business line that I support at this time. I did at one point. But the facility that takes care of that in Quebec has about 90 employees in Saint-Constant. The safety assessment business that we have where we conduct all of this research has about 2,500. So we're a bit of a kept secret. And part of me being here today is to help debunk that a little bit. So our global footprint for safety assessment, you can see here a map. Our Canadian facilities, Montreal, Sherbrooke, just up the road here in Laval, and a little bit further, Boisbriand. The European footprint that we've got, a couple of sites in France, in Lyon and Evreux, in the Netherlands, in Edinburgh, Scotland, Hungary, a couple of locations there, and on the coast in France as well, Saint-Nazaire, another site. The sites in Montreal started with our kind of foundation site in Centerville. We expanded to Sherbrooke, and in the last couple of years, acquired Cytox, which included the former LAB Research, which is just up the road here, our Laval facility. And a lot of the, as you know, here it's a small world, a lot of our leaders and employees have actually moved to Cytox at different stages, looking to pursue different opportunities. So it was a bit of a family reunion when we all came together and were able to try and start working together again. The Laval facility, I'll do a little bit of an overview just to give you a sense of our local touch, and then I'll talk a bit about the science that we're doing and some of our strategic advancements, trying to focus a little bit on technology that might be a bit closer to home for you. So our Laval site conducts GLP and non-GLP studies, has about 600 and a little over 600 employees at this point now, works with a multitude of species, has all of the support labs and pathology work that goes with it, does some work in radiation biology that we don't do at all of our sites, so it has a couple of very specialty niche areas. Also has a strong cell and gene therapy experience to offer our clients. We've got all of the support services, and you'll see some of that repeats at some of our locations as well, where we've got all of our lab sciences, analytical chemistry, immunology, immunochemistry, biomarker reviews, et cetera. The neurovirulence studies is something that has been an emerging area for Charles River. Our Laval facility has a great expertise in it, our Centerville facility as well, amongst the best in the world for that type of research. Our Montreal facility, it's sometimes called Montreal, sometimes it's Centerville, depending on how localized we want to be. It's about close to 1,400 employees now. Has been around there in the West Island since the mid-70s. So has a long pedigree and history of working in research. Was originally biorecherche or bioresearch for those that have been around for a long time. Was then CTBR. We were acquired actually by the Edinburgh facility that you saw on the map in 2001. And then in 2004, we were all acquired by Charles River and joined the broader global family there. A couple of the specialties that we have in Centerville, and we have in a couple of locations that are maybe a bit different that I'll call out. The DART and juvenile toxicology work that we've got there as a specialty. One of the things when you're looking at the safety of a drug getting to market, it's not that long ago that we had thalidomide, which was, the morning sickness treatment that actually had massive side effects and created birth defects in children. So one of the additional regulatory requirements that came out of that was to make sure to study the safety of drugs in utero, how they would affect a fetus and how they would affect young developing children, which you can appreciate, something more recently like COVID vaccines. Once we determine that they were safe, there's a whole other battery of testing that we need to do to make sure that they're safe for children, right? So that's an important specialty that we've got there. The studies that we run can actually be kind of a single dose, very short-term study up to a 2-year carcinogenicity study. So, we can have a lot of different effects that we monitor along the way and a lot of different insights that we can share with our clients with the amount of research that we've done over the years, the reference points that we've got from previous molecules and compounds that we've worked through the organization. Our Sherbrooke facility was one of the first greenfield expansions in all of Charles River globally. It was great to open that up in about 2010. It was on the heels of the global economic downturn and a big crunch time for industry as well. So we opened it up and it didn't grow quite as quickly as we had hoped it would at first, but a lovely location right in Fleurimont in the townships there near Sherbrooke. Very much close to the CHUS hospital that's there on the same roundabout. And we actually share some research initiatives with them around imaging and a lot of work with their animal models as well. So I'll share a little bit more now about who we are as an organization, and we obviously try and differentiate ourselves from other CROs. We have a huge global footprint, a huge wealth of experience, but I think it's really important with the way research has evolved that CROs have moved from being kind of a vendor, a supplier, someone that you put out tender for a study request, to really becoming a partner. And with long-term established relationships, with the multitude of studies that we've done, with the repeat work that we do with clients, we can help accelerate their work. And I'll talk a bit about what our strategies around that are down the road. So it's no longer really a one-dimensional relationship. We need to be as invested in the outcomes as our clients are, because we are all ultimately trying to serve patients, make sure that safe drugs can get to market as fast as they can, and in fact, prevent unsafe ones from getting to market as well, is equally, if not more important. So, we want to contribute to the program design. We want to make sure that we're involved in the governance. We can work with them with regulators to help make sure that everyone understands the impact that we're looking to bring to the market and to patients. Our same values, and we really talk about the 3 Ps when we talk about Charles River in terms of our priorities, and that's the patients, our people, our team members, our staff, and our portfolio. The other units within Charles River, our research models unit that I spoke about, that has the breeding of specific research models. We also have a contract manufacturing unit. We have a biologics research facility. We have our microbial testing unit, which actually is involved in detecting contamination and bacteria within pharmaceutical products and manufacturing, to help make sure that once it's even in the production stage, that batch and those productions are safe as they reach patients. We support over 15 different industries. In Europe, we've got a bit of work that we do in agrochemical and crops and chemicals. So, food chain safety is a very important part of what we do as well. We've got over nearly 2,500 scientists. We've got 1,500 members now in our employee resource groups. You know, we're recognizing very much the diversity of experience, knowledge, and background that we have across our workforce, and are really trying to champion that and enable it to bring our best foot forward. One of the things you may have seen this already, but I was able to pull out a slide that shows the typical timeline for a drug to work its way through development and the various phases of testing. And you can see it here by different kind of therapeutic area that is being targeted. But on average, we can call it 10 or 11 years. And when you think of in the US patent protection being a 20-year window, you're only potentially getting 10 years of actual protection on the market once you've got something that's been brought there. On average, right now, it's $1.3 billion invested over that 10-year window. So, you know, you're probably very familiar, but can well imagine every month, every year in the production, in the development phase can be a massive difference in trying to get some return of investment to then be able to move on and develop the next therapy or drug. So our mission in our strat window for the last while is to work to try and take 1 year out of that development cycle. And that can be done across our portfolio, whether it's in the early stage discovery, whether it's the wealth of information and data that we have from studies that we've done previously that can help you make go, no-go decisions faster. And then also, how can we enable technology to get our results, to get the data accessible, visible, collated, analyzed in an accelerated way, reported back to the client and to the regulatory authority faster so decisions can be made on the next steps. So we've really been investing a lot in technology, the way clients interact with us, the way our sites interact with each other, our departments and divisions, to leverage technology to really make that difference and achieve that goal. When you look at the stages of drug development, you know, we've got in our discovery business, which is about 10 locations globally. I used to support that from an HR perspective as well, but it grew enough that we actually carved it off and have dedicated HR support for that now. But it does a lot of the target identification, preparation of early compounds, all of the discovery stage of work that goes into it before you're ready to say, okay, I think we're onto something. Let's make sure that it's safe. And you can see right in the middle, right before it goes into the first human patients in the clinic, is where the safety assessment business sits. So it's really that critical inflection point where something is either going to move ahead or be killed. Couple of facts about the work that we do. In the last several years, we always quoted over 80%. We're now a little over 86% of all of the drugs that are approved by the FDA have been touched by Charles River at some point, either in their early discovery or in their safety testing or in the quality control services that we offer as well. All 3 COVID vaccines went through various of our facilities. Moderna, as they were starting up, you know, as a small outfit, actually did all of their work in Sherbrooke. Because Sherbrooke was a startup, more nimble site, could help them figure out more quickly what their data sets looked like, whether they were onto something or not. That was even before they pivoted to the whole vaccine space. It was when they were looking at mRNA in a more therapeutic sense. We support over 1,500 IND programs a year. We've got over 10 regulatory agencies that inspect us around the globe per year, be it the FDA, the EMEA, Health Canada, the Standards Council of Canada, SEC. Every instance of that around the world and more, we're very plugged into. We've run over 1,000 studies in cell and gene therapy, which continues to grow significantly for us. It's a key piece of the Quebec Life Sciences strategy put forward by the provincial government and the Canadian Life Sciences strategy at the federal government level. And I'm sure, you know, it's being felt in these walls for sure. And we're expanding our expertise and offerings in that area. We run over 10,000 studies a year. So when you know, a large pharma client comes to us, they've got the confidence and comfort that we've been there before. We've seen a lot of different molecules. We can make the observations early. We can do it with the right integrity and the right quality and the right expertise that we've got across our portfolio. And the smaller startups can come to us and say, we don't know what we need to do next. And we can guide them through that. We can refer to similar studies that we've done around the globe on comparable compounds or molecules, and help guide their research as they move along. And you can see here that we've got 10 cell and gene therapies products that we've actually supported getting to market. So again, a big piece of it. As I was leaving this morning, I was saying to my kids who were home with the strike that I was coming to maybe pretend to be a scientist this morning. So I won't get into too much of the cell and gene therapy specifics, because I will do that no proper service. But certainly happy to connect with some of you afterwards on some of your interest areas, and connect you to others within the organization to see if ever there's opportunities to share ideas and research concepts. From a technology perspective, and it's hard to see with the banner that's across here, I tried to get that to leave, but the technology that we've been adopting, some of it are platforms that we've just integrated into our research. Some of it are strategic partnerships where we have invested in technology organizations. Some of them have been full acquisitions where we've actually acquired that technology and brought it into the Charles River portfolio. And you can see as you look across this, some of it, at the top, Valo and Valence, AI, small molecule discovery, AI-based drug discovery. If you picture the future of safety research, if we've got this wealth of data of all of the studies that we can glean and leverage AI to help us make decisions and kind of early identify clinical signs and observations that we should be watching for in the next compound or similar compounds to ones we worked with before, this will be a critical part of taking that 1 year out of drug development. We've got a couple of others, 3D cell culture platforms, stem cell, components that are a big part of research. Deciphex and digital pathology, I'll actually touch on that a little bit in more detail, because that's one of the ones we're more advanced on and is quite cool for our pathology teams. Keyboard Medical, a partnership that we've got, implantable micro devices. That's actually, in terms of dose distribution, it's a technology that allows you to put multiple doses and multiple compounds and molecules into this single implant, and then digitally monitor as you release each of them to treat a tumor or a condition that's been identified. And you can then capture the real-time data of what the different dose levels or the different compounds are delivering as they're administered. So a lot of advancing technology in our space that has been really critical and really brought us forward. Our traditional pathology team, we've got 150 veterinary pathologists and clinical pathologists across the globe. They're a rare breed. There's about 6 to 8 of them that graduate in Canada per year, and similarly around the world. So they're a very critical resource for us, and a highly competitive landscape for us from an HR perspective, to make sure that we can keep that expertise within our family. We'll talk a bit about digital pathology. We've got over 300,000 slides and tissues that we've run through our digital pathology programming now. But on an annual basis, we can go through 3 million slides and tissues. And for a pathologist who's got boxes and boxes and slides of glass to go through and look at every tissue from different organs after the end of a study, which is really, you can do clinical observations and say, was there weight loss, weight gain, blood pressure, did their diet and appetite change, how did things metabolize through urine analysis and blood analysis. But some of the critical effects would be, it didn't seem to cause any of that, but guess what, there's a massive liver tumor that formed. So until you get to the pathology review, that's where the most critical findings can be made. And the 3 million slides to go through to find all of that, many of them where there's no findings, you can see where AI could maybe clear through the ones that are looking good and allow our expertise to focus on where we see noteworthy observations in the slides. So we have the largest group of pathologists in the world and expertise. We're very proud of that. When we talk about the challenges that come with it, I talked about the glass, the slides. Peer review, as you know, as scientific experts, is such a critical component to the scientific process that when you do a pathology peer review, it often comes from a pathologist at our client. It means they either have to fly in, spend a week sitting with our pathologist, go through all the slides and the findings, work through it again, or we have to ship all of this to the client, have them do it at their place. Digital pathology now, with ultra high resolution imaging and scanning, can allow you to actually do that through the cloud with the right technology, do it faster enable that, avoid the travel, and have more accurate findings, and build up a bank of data that you can then leverage more so going forward. It can work through helping the diagnostic features, the quantification integration with the other data sets that I mentioned, recruitment and retention of pathologists. So when we've got 6 to 8 graduating in Canada, it's a global market that we're working for. Through our digital pathology group, we've actually got one in Australia, we've got one in Spain, we've got one in Italy, where we don't have any facilities at all, but we've been able to set them up with the right equipment at home that they can look through the pathology slides and findings as if they're at the microscope in our lab, with the same quality, same resolution, and same capability to make diagnoses. It's also helped our training. And certainly you can imagine, as emerging scientists are coming out, technology is everything. The ability for them to work with the coolest tools, the fanciest toys, is certainly a differentiating factor for us in trying to get the right experts to join the organization. So how have we advanced our digital pathology? It started in 2021 with our first digital peer review. So we did the normal pathology slides, worked through it, found all the findings, worked through it, and then had to work through a validation with a peer review to say, okay, if we do that digitally, blinded, let's make sure that we find the same findings and really validate the whole concept. So we worked through that, and we were able to get it GLP accredited. We're now working through stages where it moved to then the primary review being done digitally, and then starting to move towards the AI-powered decision support that would go with it. So if we can get AI to work through it and say, this is what a normal mouse liver looks like, from all the different sections and all the different angles, put that into a bank and have AI validate the slides that are through it and say that matches what it's supposed to look like, matches what it's supposed to look like. Oh, wait, hold on, this one doesn't. Please go take a closer look. Then look at the other observations that we had from that model through the study. So really could take lots of time out of our pathology review process, improve quality, improve accuracy, and really help take us all forward. Our lab sciences division, which we've had many colleagues come to the INRS or come to us from the INRS within these groups. We've got our formulation and dose analysis groups, analytical chemistry, bioanalysis. This is the immunology, immunochemistry groups, all of our kind of in vitro services. So when you think of a clinical trial or a hospital visit, all the things that come with blood work and urine analysis and all of that, how are things metabolizing through the body? That's what this group supports. So as we're running our preclinical study, this is kind of like the clinical lab side of it that supports it. So immunology has certainly been a massively expanding factor for us. And we've got a lot of high technology in this group as well, be it our HPLCs, our ELISAs, our mass spectrometers. There's a lot of advanced technology that we use in this very expensive machinery. When we've talked to folks that have maybe gone between larger pharma or academic research, one of the attractive factors for us is that they can come to our site and we have 45 mass specs that they can get their hands on a daily and weekly basis, really advancing research and moving quickly through things that in other opportunities, maybe you don't get your hands on that type of technology quite as frequently. The last piece that I'm going to touch on is kind of that digitalization of the lab and how we interact with our sponsors and our clients. And we've developed a platform called Apollo because it was a big move. It was like landing on the moon for us. So that's where it got its name. And it's really about how do we interact with the data, with the study, with our clients and sponsors across our network. And how do we really simplify it? And it's moved now to, once it's gone live, to be as simple that our sponsor from their tablet can go in like Amazon shopping and be, I'd like to get a 6-month subcutaneous intravenous rat study lined up in Laval to see when it can start. I'll put in, I'd like it to start next month. Maybe I need a little bit more lead time, 2 months from now. What type of research am I going to be looking at it? What species do I need? Click, 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 submit it. The back end of that, then we automate our study protocol development from all of the wealth of experience that we've had of putting these together. And we can turn that around within a day or two and come back and say, we'd be ready to go in Laval We can actually do it a week earlier for you in Centerville if you'd like to, because we've got availability. Really helps people move faster with their decision making. And then moves to the next stages where they can also then get real-time data access to their study. So where in the past they would wait for a preliminary report, try and get the first sense of how is this advancing? Are there massive findings or not? What are the clinical observations? They could visit the site and monitor their study, which often at a day one, our clients and sponsors will do. They want to make sure things get off on the right foot with the, following protocols in the right way. But how do they do that without having to be there and really get access to the data? How can they monitor their study remotely, virtually as well? These are all things that we had to solve for in a faster way, certainly through COVID, where travel was restricted, on-site presence was restricted. We had to really advance a lot of these technologies, and it's actually accelerated drug development. We've kind of been forced into it, right? So real-time study data, the document management, the data visualization, all of this stuff now is available at fingertips on a real-time basis. So when we think of kind of the computational impacts of things, while a lot of what I've spoken about is more biology and chemistry, you see how much technology and computational and AI and all of those pieces are weaving their way into every walk of life. It's getting so far as even into biological research in this way. So those are the slides that I had prepared. I probably went a little bit faster than I meant to, so we might be a bit ahead of schedule, but wanted to give a bit of an overview of how our organization has evolved and the role that we play within research.
Very nice comprehensive overview of CRL, and so we now know that CRL is not just that selling.
Happy to sell some as well if anyone needs it. Yeah.
Thank you so much. And before we move on to Q&A, I would like to mention that here we have Valerie and Bridget as well, who had HR departments on different sites. So please feel free to interact with them. And we have Lab as well, who's a finance person. So if you want to talk about maybe some funding strategies, that he will be the person to talk about. And I would like to thank the professors at INRS and the students who took time to come here and see this. And it's a really global perspective of, and I see the students here are some from biotech, I guess, and some GDL students who do computational analysis and like AI and all those. So thank you for emphasizing on that, because when we try to solve problems, well, at our center, I'm a physicist from background, or chemist, I don't know, or maybe material scientist. But when we try to solve problems, we consider data as data. And then the AI takes over for everything. For us, data is data. So just to start this Q&A session, one thing we always worry about that AI might miss something important. And here is about life and death situation. For us, it's just, oh, this didn't work. Well, this is not antimicrobial. Or, so it's how do you guys take care of that how do you trust AI-based solutions? For example, we work on climate change, solutions for climate change. So sometimes there are questions about, are we producing more carbon dioxide using computers, supercomputers, than resolving the problems? So, so two of the questions that I want to ask is, how do you trust AI? Because there are a lot of researchers in the committee who do not, and there are valid reasons for that. We might miss something important. And secondly, is it really helping biotech industry? And what are the drawbacks of using AI for us? As I said, that sometimes we end up producing more CO2 than saving the planet. Yeah. So that's my first question.
Very good question. So we're not using Chat GPT for our validations here. I'm not a super adopter of AI in my personal life yet, so I'll try and speak to it as best I can. But the nature of the research that we do, the validation requirements to meet good laboratory practices and to be able to meet the requirements of the regulatory authorities, it always starts from the same place of questioning and not suspicion, but skepticism, I suppose, right?
Right.
So the ability to work through that, you know, in small slices at first is how you have to get at it. It's all about repeat validation to make sure that you get to a data set then that when you remove the AI, you have a compelling story and case to make from the data that's there. What has helped move that along a little bit faster and build a bit more trust in it, and it's still the early days in it, right? So is historically we would, you know, as technology has evolved, it went from literally typewriters making up the tables of the data to eventually into Excel sheets and gathering it, but then really being kept as standalone data sets. And where you start to get some impact and some power and some ability to trust in it a little bit more is when you've done it across 1 million data sets, right? So the more data sets that we put through it, you can build a bit more trust in it to say, hey, I had 5 studies and AI caught it right 4 out of the 5. Okay, I'm kind of sold. And I don't know if I'm going to give that vaccine to my child right away with 4 out of 5, 80%, maybe. If you run 10 million data sets and it got all but 5 of them bang on, then you start to build confidence and you start to say, okay, we've got a scientifically valid approach and method here that we can put our trust in. So it's inching away to be able to get to massive metadata type sources that would allow it to be, I think, a little bit more trustworthy.
And you probably go back to the lab and test them in the lab.
You have to do parallel, right?
Yeah.
We're not quite there to say AI is running it all. It's still very much kind of a parallel process. And the more you do that, the more you validate it, you can build the trust and the capability of it. I mean, even, you know, I made the joke about ChatGPT and the way that has even evolved from its first, you know, iterations and first deployments to where it's at now. We had a speech from an HR leader from Willis Towers Watson a couple of weeks ago that the global leadership team sat in with, and he was talking about AI in the HR space, and he put his own name into ChatGPT and said, you know, give me a bio about himself, and it started off with, you know, he's a keynote speaker, he studied in London, you know, gathered all the right facts, the basic pieces. And then it wrote 3 articles about his love of jazz. 3 paragraphs about his love of jazz, which he mentioned in one interview once 10 years ago that, oh, on the side, I like jazz. And the ChatGPT piece came out with 1 paragraph about his credentials and his, you know, his grandioseness, and then 3 paragraphs on jazz. So that's the whole part where until we get it to really be able to have enough data points where it can cut out the noise and get the real data right.
Data is everything.
It's data is everything.
All right, the floor is open for questions that you might have.
Can you use this?
Oh, yeah, I can use that. Can you hear me?
Yeah.
Okay. So actually, my question goes with the animals. You know, more and more there's some criticism, let's say, like this in animal study. Is it the real, well, ethical reason? Sometimes we see difference between the animal testing and what is actually happening in human, basically. So I'm just wondering, have you started to incorporate new methods, new models, you know, like all the organoids and so on in your research?
So I was hoping to get a bit more intel from some of our scientific leads on some of the work that we're doing there to weave into this. The technology around some of that is probably a step too far for me to be able to speak to with any accuracy, but I can tell you a little bit about what we're doing. So from an animal research perspective, you know, the 3 R's, which is a very common principle in animal research, we've actually adopted it from Charles River, a 4th R, which is responsibility. So we're really trying to look at how can we decrease the number of animals used in research? How can we minimize the impact to them? How can we, you know, how can we try and convince our clients and regulators that if they've done, you know, historically they've had to use 200 mice for this study, do we have enough data now that we've worked through that we can say, you know what, 150 will probably give you more than enough data for what you need to work with? So it's a long-term journey because a lot of the regulators that are in place that want to make sure that these things are safe have come from a history where this is how you do it, and you need to have this many species. And so there's a lot of work to, like the AI story, build up the trust and the data to say that actually it's fine with this many samples to be able to work with. Now, the technology side that comes into it is the one that we talk about a fair bit, is like an organ on a chip. So when you have enough data sets and enough intel and background around how an organ functions, and you can kind of create organoids and those types of things. You know, liver on a chip is one of the ones that we've been trying to work through in advance. So it's an important part of what we want to do. It will accelerate the research if we get it right, and it will also help improve the animal welfare and animal usage kind of component. It's a very delicate one. It's one that we've, like I was saying in the early days, we kind of weren't sure, how do we talk about this? It had a very kind of questionable or negative view to it, but we certainly hold ourselves to the highest global standards of animal welfare. And you know, we try and now talk about how critical it is to be able to get treatments and medicine to people. And I think through COVID, we saw that, you know, something that's going to be deployed globally, like the COVID vaccines, if that's not very, very safe and gone through the right animal models, how are we going to solve for this, right? So I think it helped bolster some understanding of why we need that, because I don't think too many people were saying, that sounds good, try me first. We need a little bit of testing to go with it at this stage, but certainly it's a priority for Charles River to look at alternatives.
Actually, I have a follow-up question, but the other way around. So, because I'm interested in immuno-oncology, and you mentioned safety. So when we think about all the immune-related adverse events, we need humanized mice, actually, and we cannot do it on organ on a chip. So where are you in this sphere? And
So humanized mice is something that I hear talked about a lot from our scientific counterparts. It is something that we do work on, we do develop. You know, our research models business has, you know, started with kind of transgenic mice and immunocompromised mice and models. But more and more we're seeing that, you know, kind of human-based models are becoming more and more important and relevant, especially as you look at rare disease and very targeted treatments and therapies, especially in the oncology space. You know, one of the examples that I've heard talked about, and you're probably more familiar with it than I am, but to be able to take a bisection or a biopsy of a tumor and kind of implant it into an animal model, and then you can test therapies and treatments through that to be able to see, okay, what's going to work before we go back and try and work through our menu of possible treatments with this person and this human life. Let's try and find out how we can get it right the first time in this humanized model before we then go back to treatment. So I think that's something that we're working towards as well. But you're probably a little bit more advanced on the science side of it than I am.
Maybe I can ask a non-scientific question. Like you showed us these beautiful slides on CRO client interactions. So can you just give us a few examples of partnership that we can develop with?
So some of the partnerships that we've got, you know, we work with all sorts of biotech and pharma. I had mentioned the Moderna example as they were kind of starting up, but we've got examples where we'll often kind of co-author innovative, you know, methodologies, work through new routes of administration or observations. Some of the work that we've done with, as an example, in Sherbrooke, the partnership that we have with the SHUS, where we can take, you know, radioisotopes used in the imaging in animal models, and the facilities are literally side by side, where you can see, here's the research that we're doing in an animal model from Charles River to treat this type of tumor, see how the advanced imaging can track the response to the treatment. And it's literally in the same lab and adjoining area where the patient's imaging is up and being looked at kind of in parallel and simultaneously. So we've got examples like that we leverage. I could probably think of some others where you know, it really is about you know, rare diseases where we've had you know, one of the stories was, and you can see it online, there was a young girl, Mila, that we did some research for a rare genetic disease that she had. And the client literally was working through animal model research with us with every iteration, but as they would prepare individualized dosing for Mila to try and respond to this terrible disease that she had. Interestingly, when you get a story like that and you put a human face and name to it, when you talk about 10,000 studies and 85% of the drugs, that's one thing. But behind every one of those is someone's brother, sister, mother, father, child. It really kind of compels people to make sure the work that they're doing is right. And it actually had the staff that was working on this start fundraising for Mila and her family, really take some kind of ownership of helping them through what they were dealing with. So we've got different ways that the partnership kind of comes together from a scientific, a business, and a very human side.
Just the microphone. Okay. Can you hear me? Okay, great. Do you guys do let's say a collaboration work, for example? And I'll give you an example. We have a combination small molecule synergy drugs for COVID, but we need to advance them. All right. My studies, development studies are too expensive for us. Right. Is that something that you guys would entertain?
So from what I know, when we've had instances of that falls a little bit more into our discovery non-GLP world, where it's a little bit faster, a little bit earlier stage. So it'll kind of work through some of those pieces. It's at a different cost structure as well, typically, because people are kind of in your situation. And we've got facilities for that, around the globe as well. Worcester, Massachusetts, an organization that used to be called Agilux that joined us about 7 or 8 years ago. We've got a couple of hubs that will really work through that. We're expanding a different model that can really help that collaboration and give space and capacity to advance that kind of more quickly, maybe on a smaller scale, that we've called Cradle, which is our Charles River Accelerated Development Lab. And we've got a couple of those now dotted around, usually biohubs, around the world. We're actually in early stage conversations to see if there's one in the Montreal area that we can set up. It actually falls under a different business unit, so not something I can speak to with great authority. But we're looking at now standing up some Canadian presence of these accelerator labs that could hopefully partner in those types of instances.
Thanks.
Hi. Do you do any, let's say, quantification of analytes, let's say immune cytokines or chemokines or various types of molecules found in the blood? Do you do multi-panel analysis of these types of things?
Yes, all of the above.
All of the above.
I've sat in many discussions where we've looked at cytokines, the hematology analysis that goes with it, the panels.
But do we have to do the actual study in your walls, or we can just provide?
We do it as a standalone as well. Yeah,
Okay.
Yeah, and in fact, a lot of our clinical work within the lab sciences division and within our clinical pathology division and hematology, all of those areas, we actually have a large standalone business where it's just looking at those panels and those pieces.
All right, because we do quite a lot of that, and we send our samples to Calgary to do that. So I don't know if it would be cheaper to do it here, but
certainly something we can talk about afterwards.
Checking anyway.
I don't have any business cards with me today to follow up, but maybe we can catch up afterwards. I can grab some of your details and pass.
Is it the same for pathology, let's say slides or organs that we might have, in banks? Is that the same? You can process tissue or slides that we have,
we have
without doing the study in.
Yeah, we have a strong standalone business for that. And we do that even kind of within our safety assessment facilities and labs, like I mentioned. But we actually have 3 locations across the US we call it our PAI, Pathology Associates business, that is uniquely that standalone pathology and clinical pathology review. And yeah, certainly something we specialize in.
I have a question regarding microbiome. Have you developed
Sorry?
microbiome
Okay.
biotherapeutics? Do you have, I guess, in your portfolio something about that?
We do. That's a bit more of an emerging area for us. So I probably can't speak to it with great detail, but it is something that certainly our scientific expertise, scientific experts and study directors, scientific groups are always talking about. Right now with our clients and with industry, when we are setting, our 5-year strategies, we look at, okay, what are the areas of focus that we'll need to really support better within research, and that's one of the ones that I hear talked about a fair bit.
Thank you, Ed. It was a very nice talk, and I'm happy to get to know CRL a little bit better. I was wondering, you're an HR person. I know we have other HR people here. What sort of expertise or what sort of profiles are you looking from our graduates? Are you really looking, in Quebec? I'm talking locally here because our graduates, generally they come from abroad and they want to stay, they want to integrate. Those that choose to stay here, and are you looking for kind of just basic training, as a doctoral student, research aptitudes, or are there specific skills, or are they completely reformatted once they arrive at CRL?
That's a fun expression. I'd say that, the research foundations fundamentals are absolutely critical, right? Scientific rigor, integrity, quality. I mean, that's everything in the work that we do. So that base is everything you need at the beginning. One of the things that we find is something that we need to maybe work on a little bit more in the early stages is, what does it mean to do all of that in a regulated space under a GLP umbrella? So that's something that we spend. That's the formatting that we do a lot in the beginning. I wouldn't say reformatting, but maybe formatting. So we do put a lot of focus on that. What does it mean? What rigor is required to maintain those standards? Not only to meet accreditation, but to conduct the highest level quality research that we can. So that's a good piece. And then what becomes a little bit different is you're doing the research for the research, but it's also, you've got a sponsor and a client that is incredibly invested and demanding around what's being done. So the ability to do it, I won't say under pressure, but with those parameters, with other stakeholders as involved as they are, as committed as they are, requires a skill set as well. So sometimes, how do you go back with your scientific findings in the right way as a partner to be able to say, here's what we're finding. I need to maybe challenge some of what your original hypotheses were. I'm maybe giving you some bad news about what you thought was going to work. And how do you do that in the right constructive way, or to maybe take it down a path where you are seeing some positive findings and say, maybe we should redirect the research down this channel. So that's an important piece. And then more and more, technology, the ability to understand the pace of which we can move things through the right instrumentation and that with the right speed is probably things that we focus on.
Okay, so it's extremely broad kind of expertise or general skills that you would acquire during a graduate studies. One of the reasons why we had this meeting here was that we took for granted that you would be more interested in people with biotech profiles, right? Is that true? Because we also have data scientists here from EMT that that they traveled from Varennes. We have material scientists that working with inorganic things, which will never go into a mouse or an animal or a human. We have physicists, we have engineers.
You think of medical devices and prosthetics and all of that. I mean, there are pieces of material design that will fall into our space, absolutely. And data scientists, as we're moving our digital advances forward more and more, we've now got crews of data scientists that we've hired over the last 2 and 3 years that are specifically, in that field of expertise that we wouldn't have looked at or needed years before. So all of those areas that, more and more the things that we think are outside of our scope are falling into it.
I think that's the point, because
along with AI and material science, which can fit to any data, I guess, with a little bit of background knowledge. We also have researchers at EMT Center that are developing devices or new technology, for example, ultra-fast imaging, which is very unique to our center, and we are the first one trying it. So that could be really a great fit as well.
So one of the big things with our digital pathology, right, is we need to scan all of those slides. We need to get them into the imaging to be able to look at. When again, I don't know the exact mechanics of what you're describing, but ultra fast imaging, if that accelerates the processing of the digitization of these slides and images, I mean, that again, things that sound like they're outside of scope come in scope very quickly. It's a great example. Yes, Knife.
And I'm going to ask a question on the behalf of the PhD students who are, of course, super smart, and they're creating skills in research. But what would be your advice if some of these students want to move away from academia and go into commercial landscape like ours? Personally, when I did that, it was a little bit of a struggle for me. But what would you suggest them, and see if you can include the skills that they are not developing right now that they will need in commercial environment if they were to make a move?
So that transition, can be difficult, like you said. I think what we try and do when we have people come into our organization from academia, some of it is the regulatory pieces, the GLP. I think the understanding of the ecosystem and the infrastructure around the piece of work that you're doing is probably most critical. So being able to understand, where does this fit into the bigger picture, the small part of the drug development or research that I'm doing, to be able to connect that to ultimately patient impact, or the data system that it's going to go into, or how does that then feed back to the sponsor, and how do they make their decisions? How does it then, eventually there's a commercial impact to this as well, right? How does this how does this turn into a viable business solution? All of these things are probably the extras that go beyond kind of the academic research that are there. So I think an ability to get some exposure to that is probably most critical. One of the things that I think would differentiate a CRO and Charles River for sure is the iterations and the pace with which you'll get exposure repeatedly to these things. So a chance to get an internship or to partner with, and we're very open to those relationships. Chance to build on that can probably open your eyes faster than historically, maybe a smaller volume of research being done in academia, or even in large pharma. The example that we had of someone who went into large pharma and was only able to get to work on their one molecule with their one instrument, for a very long time. Working with a CRO, getting some time with them can really help make that transition a little bit faster. And I guess the other skill to really add to it is the ability to collaborate and work with others, right? When we talk about this, the process of a study working through us, from someone who's reaching out to the client or scanning a territory to see what academic researchers are getting close to something and how can we support them, and what does it mean to get them familiar with our organization. Okay, I need to then connect them to someone who speaks their language around a study protocol design and can really help focus them. And then they need to get the study scoped out, and how many technicians and how many mice will we need, and how long will that study take, and how fast can we get the report done and turn around to them. Really understanding the kind of supply chain, if you want to call that, or the value chain of what we then deliver ultimately as data and insights at the end. That collaboration piece is everything, especially in our global footprint. The ability to be able to connect with others, effectively is really critical. And that's, in some ways, it was challenged through COVID as everything was more isolated and more virtual, but our collaboration also went that way. It became more virtual, it became more instantaneous. So I think that those real life learnings and skills, if you acknowledge them, you can actually apply them in work proactively.
Me again. We approached the aspect of data analysis kind of from the results and the product sort of perspective, but I'm seeing these graphs here, and as a supervisor of students, I like the the immediacy of having the results available kind of no matter where you are. How we have professors that work in cybersecurity, and we have graduates that emerge. And how is cybersecurity handled? Is it handled internationally? Is it handled locally? Are there employment opportunities there as well?
There are certainly employment opportunities. It is, as you everywhere, uber critical. Cybersecurity, we think of it in our personal lives, but you think of how valuable this data can be, right? If there's a reason that even in our facilities, we don't name the clients that are conducting this study, because someone might be working on the same thing, running a study on a similar treatment right next door. And we have to protect everyone's interests in that way. The value of this data, I'm biased. I've been with the organization for as long as I have, and I've obviously highly committed to the work that we do. But the value of this early stage data, can be incredibly valuable. And the protection of that is top of the list with our clients. So the ways in which we ensure that, our whole division of cybersecurity has ballooned, rightfully, because the challenges around it are only growing by the minute. So certainly there's opportunities in that space.
Even locally?
Yep. Yeah. The nature of our organization right now, we've got pathologists in Australia reading slides, right? So, it could be someone based here. They will likely be working a lot with our data centers and our international footprints, which we've got around the globe. But if you've got the expertise, we'll do it from anywhere.
Any questions from the students, the AI students? No? No questions? So I'm going to give you an example for this student. He worked with Google to identify map or bridges throughout the world using AI. Okay, that was his project. Now he's working on chemistry to identify a solution for climate change. So that's how the AI students travel to one stream to another stream. So when I am selling the idea of them to know about CRL, that's my pitch. Always. That you can apply your skill, and specifically for PhD students, I think it's the level, the pressure under which they work, and the way they are doing something that even professors don't know. Probably you will acknowledge that, because sometimes they are the ones who are
truly at the innovation stage.
By the end of their PhD, they are the masters of what they did, even though if it is a small area of research. So it's good to know that the skills that you just mentioned, they are very, very important when they step out of academia, and which sometimes are ignored in the process of doing PhD.
Yeah, when I hear thinking of using AI to scan the globe to identify bridges, I think of scanning an organ to identify certain cells, or within the cells to identify certain effects or abnormalities, right? Like it's worlds apart, but so close in the same time, right?
Again, go the other way. So you develop this multimodal AI and all this data sets. How much of that becomes public at the end to advance science? Can a researcher access all the CRL data sets and interrogate drug interactions or toxicities or what have you?
So from a kind of free public perspective, we're not quite there yet. We're still kind of building it all, right? But what we're trying to do is to make the case for our clients to want to leverage that and use that and say, beyond this study that I'm trusting you with to run, I'm placing it with you because of all of that background data and intelligence and previous analysis results that you've got to be able to influence the research, that this piece of research that I want to do. So it's something that we're, as we're building out these tools to better capture that, that's part of what we want to be able to offer our clients. That it's not just about doing this study to the best standards, as with the best pace and the best results, the fastest results, but it's also surrounded by all of that expertise that you can draw on. That's one of the things we want to differentiate ourselves against the competition.
But it's more going back to science. Do you have a Charles River models that model actually special need patient groups? For example, these groups that are often excluded from clinical trials. Let's say in the immuno-oncology sphere, patients with autoimmunity are excluded, or elderly patients, or what have you. Do you have mouse models that can actually address these special need questions?
You're testing me on that one, so I'll answer it a little bit, because again, those models are outside of the business unit that I'm most exposed to. But we do certainly, through either the transgenic models, the immunosuppressed, immunocompromised models, they are usually done, specifically for certain kind of categories or special needs, as you described, where we will purpose breed and develop the models to be able to assess exactly that need. So we do have pockets of it. That's probably as far as I can speak to it.
There are no more questions? Then we are right on time. Let's thank our speaker once again.
Thank you for the invitation. It's been a real pleasure to be here.